Hi, this is Dr. Cisse covering anatomy and physiology. I promise you guys that I want to do a little review for our exam coming. So I wanted to cover uh, the muscle system chapter. What I want to do first is to present the anatomy of the muscle, right? And then go into the physiology. We said that the skeletal muscular system includes not only the muscle as a tissue, but the connective tissue that surround them, like the membranes or the tendons that come with it. Let's present first the muscular system as a system. Like any organ system, we say there's an organ system made of organs. Each organ is made of tissues. Each tissue is made of cell, and each cell will have organelles inside of it, like the sarcoplasmic reticulum, the nucleus, the Golgi apparatus, the different things. It is the same thing with the muscular system. It's going to be just a slight difference. When we talk about cell in general, when we present a cell, we put a little circle, we put a little ball inside, like a nucleus, but in a muscle will be different. Why? Because the origin of the muscular tissue itself. The muscular tissue came from the combination, the fusion of many cells, not different, many cells, many myoblasts, right? The blast, remember that is site, that fuse together to become one long cell. That's why it will have a lot of nuclei. Normal cell will have not one nucleus, but muscle cell will have many nuclei. That's the first difference. The second difference is the shape. Usually when we present a muscle, it looks like a little circle, a little thing, but here it's like a fiber, like a thread or something like that. So for that reason, it will be different. Now, as a muscle fiber is a cell with many nuclei. When you take a lot of cells, you add them together, you form a tissue. In a muscle, the muscle fiber, let's say this is one muscle fiber, a second muscle fiber, a third muscle fiber. Now, let's talk about what is on this muscle fiber as a cell. The muscle fiber, because it's a cell, it has a membrane, and that membrane is called the sarcolemma. It's just the membrane of the muscle cell, sarcolemma. Then the inside, the cytosol, like the water inside the cell of the muscle fiber, is called, usually we call it cytosol, but in muscle we call it sarcoplasm, right? Like the cytoplasm, sarcoplasm. Then we say every cell will have organelles. The organelles inside the muscle fiber also include the mitochondria, the sarcoplasmic reticulum. Remember, in normal cell we call it endoplasmic reticulum. Here we call it sarcoplasmic reticulum, right? SR, right? Sarcoplasmic reticulum. Now, and then one of the structural organization of the muscle fiber that makes it different from other cells is that the organelles are not just in random thing. They're organized as a myofibril. So every muscle fiber will have myofibril inside of it, myofibril. And each myofibril is made of filament. We call them myofilament. And the myofilament, we got thick filament and thin filament. Now the thick filament is made of protein called myosin, okay? And the myosin head are the one that will move to make the muscle move. And the thin filament, we said, the thin filament is made of protein called actin, right? Because they look like a little circle, like a little ball, globule, we call them G-actin, which means globular actin. So it is the connection or the interaction between the globular actin, the G-actin of the thin filament, and the myosin of the thick filament that will make muscle move. Now, how does it make it move? First, let's talk about all the players. If you look at the myosin, so I'm going to erase this and put the basic unit of muscle fiber into detail. Let's say this is the muscle fiber, like this, and then each one will have myofiber. And the myofiber will be made of thick filament and thin filament. The thick filament, we say, is a myosin, and the thin filament is actin. Now, the myosin will have a head. That myosin head has to touch the G-actin of the thin filament. But I'm going to make the G-actin here. But the G-actin has a specific space, a specific area the myosin can touch. That specific area is called the active site. And the active site has to be open for the myosin head to touch it. But what happens is that the active site is not always open. It is covered by a ribbon, by a protein. That protein is called tropomyosin. Tropomyosin. Tropomyosin covers the active site of the G-actin so that it won't be exposed. Why? Because you don't want to contract your muscle all the time. You want to relax it, right? During relaxation, the tropomyosin covers the active site. But if it covers the active site, there got to be something that's holding it in place, like a padlock. So we have a padlock here that kind of holds this in place. That padlock is called troponin. Troponin. So troponin basically locks down the tropomyosin on the active site of the G-actin so that it will not be exposed. But what could open the troponin to expose the active site? It is the calcium. So calcium is like a key that can open the troponin so that the tropomyosin can be dropped to expose the active site on the G-actin of the thin filament so that the myosin head, so I'm going to pretend this is myosin head, can touch the active site to move the thin filament across the thick filament. But where does he move it to? He moves it to the middle of the basic unit of contraction of muscle called the sarcomere. What is the sarcomere? The sarcomere, it is the space between two lines called the Z lines. So we got one Z line here and one Z line here. What is between the Z line? We have the thick filament and the thin filament, right? So the thick filament doesn't move. It is anchored to the Z line by a protein called titin. It is the protein made of elastic fiber. So it's called elastin. So it kind of connects the thick filament to the Z line. But the thin filament is connected directly to the Z line, such that if the Z line is moved, the thin filament will move. But the middle of this unit called the sarcomere, right, is called the M line. The M line is the midline. The M line divides the sarcomere into left and right. So they are made of protein that hold things together here. So during contraction, what happens is that the calcium is released inside the myofibril, and the calcium comes and unlocks the troponin, and when the troponin unlocks, it drops the tropomyosin off the active site of the G-actin on the thin filament to expose it so that the thick filament can have the myosin head touching it on both sides, pulling them towards the M line in the middle of the sarcomere for muscle contraction to happen. But where does the calcium come from? The calcium is stored in the structure called sarcoplasmic reticulum. It's the same thing as the endoplasmic reticulum, just different name in the muscle tissue, right? So the endoplasmic reticulum in normal cell is called what? Sarcoplasmic reticulum, or SR. What is inside SR, sarcoplasmic reticulum? Calcium. Now, why do we need calcium? Calcium is the key that can unlock the troponin so that it can drop the tropomyosin to expose the active site of the G-actin on the thin filament to make it open such that the myosin head can touch it and pull it towards the M line. But what is on the myosin head that gives its power? The myosin head is energized, is activated by energy. That energy is called what? ATP. So ATP will be used on the head of the myosin to energize it, to make it open so that when there is calcium, it can drop the tropomyosin off the active side of the G-actin so to energize it. In order for the myosin head to do that, it has to go around the active site, hold it to form a cross bridge and pull, we call that the power stroke, to the midline. But what will make the calcium be released from the sarcoplasmic reticulum? It has to be through a nerve impulse. Why? Because muscle and nerves are excitable cells. What does that mean? It means that they can be excited. You can put something on them that will make them do something. They can work and do something. So any muscle that works has to work under the command of a nerve. So the connection, the relationship between the nerve and the muscle, that connection is called what? The neuromuscular junction. The connection between the nerve and the muscle junction. Now, what is it inside the nerve that gives it the power to energize, to activate the muscle? The nerve is an excitable cell also. The nervous system's job is to communicate. If somebody tells you to raise your hand, the message comes to your ears, go to your brain, get analyzed. It then go to your brain stem, your spinal cord, and your spinal cord will send nerve. And the axon, like one side of the nerve is called the axon, those come together as a fiber to go to a specific muscle cell. That end of the nerve will secrete, the nerve will secrete, the neuron, the cell of the nervous system will secrete in acetylcholine, which is the neurotransmitter. That acetylcholine is secreted at the end of the neuron. So I'm going to draw the neuron here quick. So we have a neuron from the nervous system, and that neuron will secrete acetylcholine, right? So the acetylcholine will bind to a receptor on the muscle fiber, and that receptor will open the door so sodium to come in. Because sodium is positive and the inside of the cell is negative, positive into negative will create electricity, basically, like electrical impulse. That electrical impulse will go through a tubule. That tube is called T-tubule, T for transverse, because it goes in a transversal way. And that T-tubule will connect with the sarcoplasmic reticulum where the calcium is located. And that calcium release will bind to troponin to drop the tropomyosin of the active site of the G protein of the thin filament so that it can expose its active site, right? In order for the myosin head of the thick filament to be able to touch it, to form a cross bridge and to pull it. We call that what? A power stroke, to give a power stroke, to make the muscle move, the fiber, thin filament move across the thick filament so that the sarcomere can be shortened and the muscle can be short. Now, where do you get the energy to make your muscle move? It depends on the situation. In normal condition, your muscle will use the energy, the food you eat, and the air you breathe. You got oxygen, you got food, like a carbohydrate, protein, nucleic acid, and fatty acid, like lipids. Now, they go to your body, and your muscles use them. And it depends on how far you're going. If you're just a regular situation, your muscle will use just the regular ATP energy in the muscle. But when you exercise, depending on the level of exercises, the energy can come from just phosphate, just take phosphate and add to ADP. Because when you hydrolyze ATP, that's triphosphate, you break it down to pieces. ADP, adenosine diphosphate, and the inorganic phosphate. Sometimes when you exercise at the beginning of the exercise, your body will pick up those phosphates and add to ADP to create energy again, right? But if you exercise for a very long time, like a very longer, your body will actually break down your fat, or it will go to the glycogen inside your body, break it down to make glucose. So that's going to be different kind of level of using energy based on the need of your body. Now, what about the muscle fiber itself? Remember we said the muscle fiber is covered by sarcolemma. On top of the sarcolemma, we have what? The endomysium. Now, if you take one muscle fiber, it is covered by endomysium. But if I add a second muscle fiber to it, right, and I add a third one, all this will be a fascicle. So the fascicle is covered by the perimysium. And the fascicle is like a tissue, right? Then if I take a lot of fascicle together, I form an organ like a bicep brachii, like rectus femoris, or quadricep, or tricep, or bicep femoris, or bicep brachii. So all those muscles are organs. They will be covered by epimysium, right? And then when you take all the muscle together, everything else is covered by the fascia. So now, let me see if we covered everything for our lecture. exam coming. So we talk about iso. Okay, let's talk about a different type of muscle contraction. Now, if I wanted to contract my muscle, I want to hold something. If I put my hand under a table that's too heavy that I can't lift, as I'm holding, I'm building a tension in my muscle, but I'm not making the length change because my hand is not flexing. Because there's no movement, it's just me trying to hold up to something. We call that the isometric. Remember the metric system? The isometric, meaning the meter is iso, is stable. I'm not changing any length. I'm just holding something heavy, like that. But if I'm picking up something, I'm just holding, then my muscle has to be shorter to pull me. The length will change, but and the tone will be maintained. We call that isotonic. The tone is the same, but the length change. If it changes by shortening, we call that concentric. If I'm dropping something slowly, but I'm still holding something heavy, like that, the length is getting longer, but I'm still dropping slowly. That's called eccentric. So it's gonna be isotonic, right? The same tone. Eccentric make it longer, concentric make it shorter. But if it doesn't change length, it's called isometric, right? So let me see. All right. So we also talk about the strength of the muscle, which we say depends on the size of the muscle. Big muscle will hold bigger stuff. Small muscle cannot hold big stuff, right? And then we'll talk about the neuromuscular junction, talking about how the nerve talks to the muscle. The nerve and the muscle unit is called the motor unit. Why we call it motor? Because the nervous system talk two ways, like highway. You know, you go forward, you come, you go back and forth. It's the same thing. Your muscle receive information from your nerve through your sensory neuron. But for them to do something, they have to respond by a message from a motor neuron. The motor neuron is the one that makes it like a motor, like a machine, makes you work. So our muscles receive the motor neuron that secrete acetylcholine to work, right? So the reception of the information is called sensory. The response to that sensory information is called effector, like it does the job. It kind of responds to the situation, right? And then we talk about why muscles are excitable, because the inside of the outside being different charges, the entrance, the coming of sodium positive ion into the muscle creates exchange, it switch charges, right? It switch charges from negative to positive. So switching charges from negative to positive create electrical power, basically. We saw that the cell has been depolarized. Why? Because it was negative inside and positive outside. They used to be polarized. It should be different pole, negative and positive pole. But when you add a lot of positive inside, you kind of depolarize. You remove the pole you make it the same thing, basically, right? So let me see if there's talk about everything else. We talk about the myosin. Yeah, I think that's it. So you need to know the anatomy of the muscle tissue. You need to know the whole muscle is covered by the epimysium, the fascicle covered by the perimysium, and the muscle fiber covered by the endomysium. But if you go under the endomysium, you need to know that the sarcolemma is the cell membrane of the muscle fiber. The sarcoplasm is just the cytosol, the cytoplasm of the muscle. It has mitochondria, and then it has organelles. One of them is sarcoplasmic reticulum covering the myofibril. So now let's talk about the organization from small to big. From small, the thin filament is made of G-actin. They come together to form the F-actin. The thick filament is made of myosin. The thick and thin filament together is called the myofibril. If I take a lot of myofibril together, I form myofilament, right? So thin filament, thick filament are all filament, but when you put them together, you form the myofibril. If I take one myofibril, two myofibril, three myofibril, many myofibril, I form the muscle fiber. Then if I take many muscle fiber together and mix them, I form the fascicle, which is the tissue. And then many fascicles together, I form the muscle as an organ. Like, for example, rectus abdominis or deltoid muscle or pectoralis major. Let's just practice the name of the muscle, right? Okay, frontalis, right? Occipitalis, temporalis, temporoparietalis, masseter, buccinator, orbicularis oculi, orbicularis oris, rizoris, right? Mentalis. We talk about levator, right? The one, levator, labii superioris, depressor, labii inferioris. We say depressor anguli. Then we come to the neck, platysma, and then trapezius on the back, sternocleidomastoid muscle. Then we come to the pectoralis major and pectoralis minor. Then we come to the rotatory cuff muscle, rotator cuff, right? Supraspinatus, infraspinatus, subscapularis, and teres minor. Then the next muscle, we say the deltoid, teres major. We come down to the arm, bicep brachii, brachialis, coracobrachialis, back, tricep. Then we come here, we say flexor carpi. Oh, first of all, we talk about the brachioradialis, and then we say flexor carpi radialis, flexor carpi ulnaris, flexor digitorum, right? And then we say palmaris longus, palmaris brevis. We come to the back, extensor. We say extensor carpi ulnaris, extensor carpi radialis, extensor digitorum, right? And then we say that's it. We come to the abdominal region. We say external abdominal oblique, internal abdominal oblique, transverse abdominis. We say rectus abdominis, right? On the back, between the two scapulae, we say rhomboid major, rhomboid minor, and then we say the erector spinae muscle on the back, semispinalis capitis, we say spinalis, and then we come down to latissimus dorsi. We come to the leg, ilium, we say iliacus, the one from your lumbar to your ilium, we say psoas, remember the psoas, P. Then we come to the leg, we say the rectus femoris, we say vastus medialis, vastus lateralis, vastus intermedius. We came to the back, we say bicep femoris, we say semitendinosus, semimembranosus. We came down, right, to the calf, we say gastrocnemius and soleus, we say tibialis anterior, tibialis inferior, and then fibularis. Basically, those are the muscles we need to know. Maybe I forgot some muscles, I'm not sure. And then we talk about the diaphragm, divided in thoracic from abdominal. And then the muscle, we talk about their functions, which one makes you flex. The flexion will always go on this side. We see the leg is different. Extending your knee will be on top here. The back will be flexing. At the same time, flexing the whole leg will almost be like extending your knee because it's also lifting your leg. What else we need to talk about? We talk about the length and tension relationship, right? Power stroke, head. Okay, we talk about all that. We talk about acetylcholine. We talk about the T-tubule. We talk about how we use glucose to make energy. Phosphagen system, how we go to the trash can. Remember during tornado? We go to the trash can and find food. If we were informed, that's called the phosphagen system. Your body's kind of smart. When you don't have energy, it finds a way to make up the energy. That's so we say the muscle is voluntary. I think that's it. If you know all that, you can make it. So just come ready, practice the name of the muscles, practice the name of their function, how they make you move, right? You know that by knowing the instruction and the origin. Remember, usually you go to your origin, you pull the instruction to the origin, that's that function. So if you're given two different parts, if this is the origin, this is the instruction, usually let me come in to the origin. So to understand. So know the troponin is just a padlock. Know the tropomyosin like a ribbon covering the face, the active site. Know that the calcium is in the sarcoplasmic reticulum. It is released by the nerve impulse that comes through the T-tubule, which is done because acetylcholine is secreted by the nerve that will open the receptor opening to let sodium into the muscle fiber. Sodium is positive, it goes inside to make it negative. To balance off with the negative. So the coming of this positive into the cell will remove the polar position, like they're not going to be polarized, I mean depolarized. It's called action potential, creating power, energy that will go through the teachable and then to go through the triad into the sarcoplasmic reticulum, right, next to the triad, and then make the calcium release that will bind to troponin, drop tropomyosin, right? You know the story. All right, thank you very much. I hope you're going to enjoy this video and learn from it. You can share it with people who want to know about this, and then we go from here. Again, thank you very much, and I'll see you in class.